Greetings, programs, and welcome to INIT Hello 2026. I'd like to thank my opening act. And I am not bringing you any sort of death maze, 5,000 or otherwise. I am bringing you something called File-Ogeny, or the untitled digital phylogeny tool, named to be determined later. You might know me, I'm Charles, aka Option 8. In the past, I have brought the community such marginally practical creations such as Apple II native Bitcoin mining, a machine learning algorithm that teaches itself to play tic-tac-toe a la Whopper from war games, and an Altair emulator that optimistically runs at about 1% of actual Altair speed, including clicky buttons on the front panel. So what have I actually brought for you today? Today, I am introducing, well, I'm branching out. Instead of moderately pointless 6502 assembly or AppleSoft programs, which I'll have more of for the contest, I think, I have written a Python program this time that runs on modern machines. So far, that includes this Mac. I haven't tested on anything else, but it does have some actual practical purpose. You may now express shock and dismay. In a nutshell, File-Ogeny, which right now is a working title, it was a bad pun, and I'm open to hearing suggestions after the talk is over. Thank you. What it does is it compares multiple files to one another all at the same time. It could be different versions or different variations of the same file, text file, binary files, whole disk images, very small sequences, whatever. Any collection of text or binary data that you want to find the similarities between. And there are plenty of tools in the modern software toolkit that help you find the differences between files. You know, the gold standard is, of course, the classic, or the Unix tool diff. There are visual differs, ones that work on binary specifically, large files, multi-byte Unicode text, weird formatted things, extremely large databases, finding unique keys, that kind of stuff. So there's all kinds of ways to say how this file differs from this one in what way. My intent was to try and find something I could use to find the segments of a file that were actually the same, what they shared across multiple, say, iterations between different generations of a file, different versions of some source code or program, or what I find more interestingly, between different programs entirely. They might have some bits of shared code. My thinking was maybe I might find a block of some data or code an author might reuse in different games or different programs, a library or a standard set of routines that might have been repeated from all in the same publisher, whether it was reusing the copy protection scheme, that kind of thing. Or maybe I could identify where some little chunk of code was maybe lifted, copied, plagiarized even, or some kind of maybe a signature that you might help pin down the authorship or the date or the origin of some sort of unknown work. Was I able to do any of that so far? No, not yet. But I was able to make some pretty graphs. I'll show you more of these in a minute. And they will be suitable for printing. So if you'd like to put them on your pen plotter or otherwise, they will be available for purchase in my shop. So what is File-Ogeny? And I'm not even sure if I'm pronouncing it right. To be honest with you, anyone in the biological sciences able to tell me if that is the correct pronunciation? Yeah. I'll take your blank stares. Oh, hey, somebody actually spoke up. Great. Phylogeny, all right. Anyhow. I didn't hear any of that. That's fine. Phylogeny is the phylogeny. I'll use that. Is the study. I'm the one with the microphone, the evolutionary history, development, and relationships, and I only have 30 minutes, among species or populations. So you might recognize the output of some phylogenetic tools. Again, not sure about the pronunciation in that, as it is the process that's used in building things like a tree of life or the genetic family trees, in this case, families of crocodiles and related birds and non-avian dinosaurs, et cetera. And then using that to infer how they were developed from extinct or unknown species that you might find in the fossil record. So in genetics, this process is applied to things like DNA sequences. So protein sequences or specific genes or a whole encoded genome from, say, a bacterium or something. The sequences of DNA, the base pairs are actually aligned, usually using some kind of algorithm or AI. There's a whole bunch of them that competing best fit algorithms, and usually some human guesswork. So there's, trimming the ends and finding where things might have been misaligned, that sort of thing. And then they're analyzed. That goes to one of several different analytic schemes. The biologist in question chooses from that gives them the results that they're hoping for. Each algorithm, of course, is optimized based on different goals, whether it's looking for single base pair mutations or common transcription errors, that sort of thing, all that. It's a whole fascinating field of study that I just kind of barely skimmed the surface of. Just so I can understand what it was that I was trying to do and that it had already been done before, just with DNA as opposed to binary data. So each of those algorithms, of course, is tuned using exhaustive statistical analysis and uses things like math terms like distance and similarity, but they mean very different things when you're talking about mathematical distances and similarities and graph theory and metrics and large example data sets and things like that. My program, by the way, almost none of that is true. So the question you're all thinking, what does all this have to do with retro computing? Well, the answer is biological evolution takes millions of years and thousands of generations to sort of wedge a species into its ecological niche, where survival is based on how well individuals can find food, not become food, and live long enough to reproduce. Some of those mutations and branches and dead ends they all end up in the fossil record. Others, we may not know anything about them other than what they've left behind in the DNA of living species, and scientists can kind of find that through the sort of track changes version of the DNA record, like lines of code that have been commented out after you've debugged them, but they're still in the binary because you can't recompile. It's all kind of a pain in the ass. But software, similarly, evolves, but much more quickly, and a product or program or tool or utility or whatever will only get sometimes 1 or 2 generations, if it's lucky, to prove that it's viable before going extinct because of lack of funding or interest or being outcompeted, that sort of thing. Projects are canceled, they go obsolete before they even get released. That happens a lot, and we've seen projects and talks about that sort of thing at this conference and others. But even so, the evidence of a program, its ancestry, and sometimes its its origins and some of the secrets might actually still be lurking in the code itself, structurally, or passages that have been commented out or bypassed around, or what have you. And actual archivists that know what they're doing, not me, have recognized the importance of preserving that fossil record. And again, we've had more talks about that. And everyone hopefully understands the importance of preserving all the things: backup tapes, floppy disks, beta versions, rare one-off obscure things, source code printouts, the OCR data, that kind of thing. All of that stuff, even if it's corrupted or fragmentary, there is information in there that can be useful to maybe answer questions or fill in some gap in that fossil record, if you like. So why is this important? How am I contributing to this? Remember this, I am not a historian, nor am I a biologist, geneticist, archivist, or whatever. I just bang on the keys and make Python code in this case. But I know some people who are, and that they tell me is yeah, sure, anything helps, right? Okay. So what does it actually do? Now that I've given you the whole background and the spiel about genetics and DNA. So step one, we're going to determine how similar two or n different files are to one another. So similarity. And one of the ways we can do that is super easy is through file compression, strangely enough. So how does one use compression, a compression algorithm, find the distance sort of statistically between two files? So when I compress a file with LZW or Lempel-Ziv Welch compression, that's part of PK-zip and a lot of the zip programs and compression programs that you've run into all over the place. And even like Z-modem and things have LZW. I don't know, all the things that compress stuff, 90% of them have LZW at least as part of their, or starting point of their compression algorithm. So what the scheme does is, as it reads the file in, so starting file, resulting file, it reads it in byte by byte, tags each unique block, how large the block is, how many blocks it can identify. It all kind of depends on the implementation, but the idea is that if that block, or sometimes multiple blocks, varying lengths, if the block appears later in the file, we don't have to get another copy of it. We just say from here to here is the same as here to here. So you get a little shortcut that says, look there when you're inflating the file. So you get multiple shortcuts pointing to the same chunk of data. And those don't take up much space at all, usually much less space than the original chunk of data. So it moves on, different chunk of data that's unique for the first time, but then that happens again. So another shortcut, and eventually you end up with the compressed file, in this case, in my theoretical blah, 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 70% savings because it's only so many unique blocks repeated over and over again. This is great for things that have a lot of repeated blocks, but if you have something that is pure random noise, you're not going to get any compression because there's nothing that is more than a few bytes long that is repeated. In this case, it's just garbage, static, highly compressed something already, or just randomness. You won't get it any smaller by running it through a compression algorithm. But if you have two files that are highly complicated, mostly pure random noise, then it gets interesting because if you concatenate those two files together, they will get smaller because the similar pieces of random noise, if they are the same random noise, well, that'll get a whole lot shorter through that same compression, that all that region becomes is a shortcut. And so now you can see it's not as much as something that is a simpler, more repetitive file, but it'll still give you a compression, a measurable result. And what happens is you do that, comparing the compression results of file A and file B against the result of the concatenated version of file AB, and that'll give you a measure of how similar those two files are. The smaller the combined file compresses versus the individual files being compressed means smaller means more similar. So if the two are totally unrelated to each other, they'll be just as big as if you compress them individually. But if they are very much similar, you end up with a smaller file. This is important to the next couple of steps. So is everyone grasping? Okay. Awesome. Because this is where math happens. I usually say this is where the magic happens, but oh, sorry. Smaller, more similar. Okay, good, good, good. So in the end, we've got, this is a list of like 8 or 9 files. And this is a matrix of the distance of the first file to the second, first file to the third, first file to the fourth, second file to the first, second file to the second, second file to the third. So you see the diagonal has a distance of 1, or it's actually a reverse, it's the similarity. So the first file is, of course, 100% similar to itself. So diagonally down here, it's going to be the same, the 8th file to the 8th, and so on. But with a matrix like this, we can put it into an online phylogeny tool that will build you a tree, in this case, of different versions of the Apple IIc ROM. Now, of course, this is optimized for DNA sequences, and it's looking for things like common ancestry and using the distance matrix to build a tree as opposed to, something else. So it's unlikely that the ROM 4 and ROM 3 are descendants or siblings of the Apple II Plus ROM or whatever, but you get the idea. The format is the same. You've got a matrix of distances / similarities between a sequence of, in this case, bytes versus DNA. So who's heard of the traveling salesman problem? So we're getting back into computer science area as opposed to just math, just pure math. Great, so computer science, traveling salesman problem. Each pair of files or nodes now has an associated distance between them. So starting with the first node, we make the path to its nearest neighbor. From that, you add the path to that node's nearest neighbor, as long as it wasn't also the nearest neighbor of the first one, excluding the nodes that we've already traveled to. And so we build out a path that includes all the nodes. And this is all these are the same distance apart if you're looking really closely at the numbers, because that's copy and paste. Anyhow, so as you build those paths, you end up with a shortest path starting from point 1, and then another shortest path if you start from point 2, and another shortest path if you start from point 3. And then you compare all of those paths to find the shortest of those paths that include all of the nodes, repeating the process with a different node starting point each time. Again, maths happen. In this case, for our non-American friends, it's plural. So the shortest combination path that covers the whole list is the order of the files that we do the comparisons with in part 3. So we're not done yet. So to recap, exhaustive traveling salesman algorithm, which is to say not an algorithm at all, just brute force. And the shortest total distance is our best match ordering. So another sort of idealized situation, we've got a couple of text files here. Once the order of similarities has been established, we get to compare each nearest neighbor to it, each file to its nearest neighbors. So finds the specific region in the file, starting at the top, reads the data, which usually has a minimum size region. In my program, I start with a default of 8 bytes. It can be as long or short as you like. It's all something configured on the command line, if you like. Starts with beginning of file 1, searches for an exact match for that chunk in file 2, which could be anywhere. In this case, it's at the top. If a match is found, then it expands that region until it gets to the end of the file, if necessary. But it expands the region that it's looking for, looks for that new region size in file 2, and keeps going until it gets to a no longer matching region of bytes. It marks that region, this is my match, and then moves to the next set of bytes. So the next set of bytes, again, it could be anywhere, continues until it gets to a no longer matching region, tags it, moves on, repeat. So that's the background. Each region that gets matched is recorded with the offset in the origin file, the length of the match, and the offset in the destination file. You can say there's 100 bytes in file A, and they start at this offset, and they match 100 bytes in file B at this offset. So that is a thread, if you will. This all gets rendered into an SVG file. This is a scalable vector graphic. And that means that you can zoom in very closely on small regions, et cetera, or you could print it out, or you could get it tattooed on yourself. Whatever you want to do, it's just an XML file, really. So you can either look at it visually, or you can parse it with some other XML parsing tool. And now, let's see if I can get this over to the other screen. View, and let's see, can I stop this? There we go. And I get a mouse? No, I guess I don't. I guess I'm just going to have to do mirroring. And screen mirroring. And that extended there. Tell me if that changes. Displays, I want to display mirror. Screen mirroring. No, okay. Why does it keep showing me Philips MacBook Air? Somebody have, that's why. Keeps offering to mirror my display to somebody else's laptop is the problem. All right, so I want to choose extended display. There it is. Now that it's not, there we are. Yeah, now that it's not offering up someone else's laptop. There we are. That's much better. All right. So, yeah, so this is, where is my, I got my notes all sorted out. Oh my goodness, you got, okay. So, again, this is all the result of the process that I outlined earlier. This is 24 different versions of the ProDOS kernel. And if you look closely, you might notice that they are in roughly correct order as far as the version numbers go, starting with 1.0.8304 all the way up to version 203. There's a couple places where it swaps, gets a little bit out of order, but that is the result of the best matching algorithm. It's not alphabetical order. This is how it chose to order them based on the distance from one to the next. And because this is an SVG, I should be able, if I can switch to my, there we go. I can zoom in, and if I roll over a region, this one is particularly complicated because it's a big file and it has lots of little chunks. And I've made the minimum matching size 8 bytes. So literally like load A, store A in a thing. And so you'll get matches that kind of jump around a little bit, like this little byte somewhere up here is matched way down here, and that's the first time it appears, or the second time that it appears in this, this version over here. So these large regions, that's where the code doesn't change at all from this version 2 something to this version 3 something. But you can see where there's been some code changes here. You can see where obviously the files have gotten bigger over time. There's more and revisions, refactoring, rearranging stuff inside of the kernel. But that's the gist of it. in ProDOS. But again, that's kind of cluttered. That's 24 files all at once. And it's also going from right to left. The distance, the best match algorithm doesn't look for what order things happen in. It just says these are a distance apart. These are the closest. So I've made it so you can reverse the file or reverse the sorting. So now the latest version is over here on the right. So if you find something that obviously this was made in 1983 and this one was 1981 and this one was 1979, then, well, we can just hit the reverse sort and it reverses it. It's still kind of cluttered though. So I'm going to make the minimum byte matching size 32 bytes instead of 8. So only major, big chunks that have been changed from one to the other or that remain the same from one to the other are going to show up as threads. Again, this isn't showing the differences, it's showing the sames. So if I see a big chunk here that's the same between these two versions, and then only these little bits remain the same, that means that all the stuff in between, nothing that is 32 bytes or longer remains the same from this version to this version. So that's a big refactoring or compression or something. And you can see that there are big chunks that are rearranged vertically as well. So there's like, something that got shoved to a different place in the kernel, that sort of thing. So that's kind of a look at the interface of how it works. And again, this is just viewing it in Chrome, and I've added some stuff into the SVG. So you roll over it, you see the metadata. So interactive somewhat, but again, it's all XML. You can parse it to your heart's desire. How am I doing for time, Paul? Okay. All right. So everybody has a copy of Sabotage, right? The game that appeared on probably a dozen different games collections over the years, and every BBS had a version of it. This is 19 different versions of a game found on Asimov called Sabotage. It was on a bunch of different disks. Which ones are the same version? Which ones are different versions? Which ones are actually Sabotage? Which ones are not? And they're just named that for whatever reason. Or it's a totally different game, just happens to have the same name. So this is a quick scrape. And there's a bunch of them that are the same. You can see the boxes, the same color from top to bottom. So what I do usually is I tag the files with an MD5 hash to compare them one to the other so that if they are the same, then it just eliminates a lot of the duplicates. And so we get down to however many this is, 9 different versions. And so there's some strangeness here where like maybe somebody's got an earlier version and a later version, or somebody's hacked it to make it a little bit easier. You get infinite ammo, or put their name and logo in it instead of the original authors or whatever. I'd have to actually open the programs and find out. Some of them I did. I opened up the program, launched it, looks like Sabotage to me, and I closed it. There's no indication of version 1.0, 1.1, 1.2. It's just a game. And then this, that's clearly not Sabotage. It's called Sabotage, but I named it not Sabotage because it's roughly the right size, but doesn't have anything in common. And then this one over here, this one's very different. And then it looks like it plays the same. So maybe it's an earlier version. Maybe it's a later version. I don't know. I don't know the order, again, of the similarities and differences. It just happens to be named that on a random DSK file on Asimov. So fun things like that. I don't know what they mean. I'm not a historian, again, as I said. So this obviously also works on DSK files, whole disks at a time. So this is the Sticky Bear Math disk file. And so you can see that there are multiple things that are called Sticky Bear Math. This one is at least named revision 4. This one came from Optimal Resource. This one is, a slightly different version. This one says 1984 and Optimal Resource. And so they're all these different times that it was preserved, different times that it was ripped. Maybe it was a cracked copy that was copied again multiple times. There's, chunks of the header and the catalog that are the same, chunks of the content that are the same, and then there's this one over here that's totally not the same, but again, it looks like the same disk if you put it in your emulator in this case. So those are the kinds of things that I'm finding just randomly, again, with my own, copy of, a rip of the Asimov, or some stuff that I've grabbed off archive or things that people have ripped here this weekend so far. So this is Sticky Bear Math 2. Again, same kind of thing. All the Sticky Bear Math content is the same, but then there's some stuff in the headers, maybe different files, different trademarks or copyrights or whatever in them. And we can zoom in again and see what the individual chunks are, and then open that with a hex editor and see where those files break down. So in here, got a couple more examples. This is Copy 2 Plus, which was going to tie into Mark's presentation, including the 9.0 beta that he found, or that someone found just the other day, and where it fits in to the major refactoring / rewriting of 9 when they lost the source code, essentially. So there's where 9.0 starts. It very little remains from 8.4. And then even 8.0 makes major changes, rearranges from 7.4. And you can see that happens back here again at 5 to 6, and then 4 to 5, and so on. So this is kind of the entire history of that utility.sys file on Copy 2 Plus. And then here is the same, but for the parameters file. So if you're copying Wizardry or Bard's Tale or Wings of Fury or whatever that has copy protection, Copy 2 Plus will recognize that disk and do the things that it's necessary to do to make a copy of that protected file. And so that's in a parameters file. And you can see that parameters file starts out real small and gets bigger and bigger and bigger, and then obviously gets reformatted at some point from 6.1 to 6.5. Major change there. But all the contents there is just rearranged. Maybe they alphabetized it. And then again happens between 7.1 and 7.2. So what happened there? It's all there. It's just all in different order. And then we can see that, that 9.0 beta disk, it has exactly the same parameters data as the 8.1 copy. So 9.0 beta maybe started its life as a branch of 8.1, or at least they copied the parameters data from the 8.1 generation. So again, that was going to tie in with Mark's presentation. So for future reference when he comes, when he is back in fighting order. This is an example of what it looks like when you use the same program on source code. In this case, a program that a shell program, bash program I use called Patchomator that I wrote that automates software updates and patches. And you can see how the script has evolved over time, gotten bigger, rearranged, et cetera, et cetera. So it works on text files, really good for tracking through changes of source code, but it also works on Shakespeare. So I thought, well, how different could Hamlet be in the 1998 transcription of Hamlet from the 1603 original version versus the 1623 first folio versus, so you can see that the English spellings have changed, and some of these have, the longer ones have all the stage direction or notes for an actor or notes on the transcription itself in amongst the Horatio, how well I knew him, etc. And then the version from 1990 and 1998 are virtually identical because they're getting a modern source, and I don't know, you don't translate Shakespeare into English so much as just maybe annotate or correct typographic errors, that sort of thing. So same tool will work on pretty much arbitrary anything. In this case, also DNA sequences. So coming full circle, it'll also work roughly as well as a generic open source free. You don't have to credit them in a journal if you publish your data version of the DNA phylogeny tools, which again, I take that output and send it through this web portal, and I should get, continue, where'd my tree go? I had a tree. So if I view my tree, oh no. Well, in any case, it will make a valid, I must have just gotten dropped off the Wi-Fi. Anyhow, it makes a valid DNA based phylogeny family tree. And I know I have run over time, but let me see if I can get Keynote to take the screen back. Ah, there we go. Use cases. I'm not a historian, not a biologist. I just bang on the keys. And if you have any questions, now would be the time. If you want to get a copy of the latest version of this and instructions for running it on your Mac with Python, then that is the QR code that takes you to my GitHub. And there are some questions, asterisk, that I'm not going to bother answering, like, are you pronouncing that right? So again, I know that I have gone over time, so anyone have questions, please fire away.
So even in DNA, you've got sections of junk DNA. Have you thought of maybe putting at the front some sort of static analysis to make it a little bit context aware? So like in
the only context awareness that my program does is it strips out, well, in text, it strips everything back to ASCII. So not even just ASCII alphanumeric. So if you've got multiple spaces, so white space, punctuation, high byte, low byte, et cetera, it strips it back to just alphanumeric stuff. So all of that Shakespeare, anytime they had the special character for the long S, for instance, instead of, like congreffs, if that wasn't translated by Gutenberg into S or F, it would have been stripped out. So that word wouldn't have matched in a later version where that character was an S. The other thing that it does with binary data is if it has a string of more than 4 zeros in a row. So if it's got a sparse file that has a bunch of empty data at the end, but it's a megabyte long and it's only got 100K of data at the front that's actually data, it strips out anything that is, or basically compresses out anything that is a string of specifically zeros. So if it's a string of no ops, it's not going to strip that out because that might be just as significantly a string of green pixels if you're looking at an image. So generally, if it's zeros or nulls, it's going to be a sparse file, and so it strips those out, only compares things that are not null at that point. As far as junk DNA goes, that's probably the closest I can come up with.
Probably should cut this off here.
Yes, we're cutting it off now.
Because we want to leave a little bit of time for people to go do what they need to do before we come back to the next talk, which will be Jeff Weiss talking about neurons. So you have 5-ish minutes.
Thank you. So if you do have any more questions for me, please come find me. I'll be here all day and tomorrow. And don't forget to tip your waitresses.